In this video, we are going to be looking at how DNA is synthesized inside the cells, and we're also going to be looking at how other biomolecules are synthesized starting from DNA. So first, we are going to be discussing the process of DNA replication. So DNA replication is an event that occurs when cells are dividing. Now, understand that DNA is going to be copied to then produce this genetic material that is going to be utilized for new cells in order for them to make other biomolecules. So again, the process of DNA replication, number one, we're making more DNA. This process is known to be a semi-conservative process. So what this means in a semi-conservative process is that when DNA is made or replicated, a parental strand will be base pairing with a new strand of DNA. Okay, so if I do this by drawing, let's say that our parental strand, meaning the DNA that is going to be replicated, let's say that it is these two strands. When this parental strand let me just write this a little bit more clearly. This parental DNA undergoes DNA replication. These parental strands are going to be base pairing with brand new strands of this DNA that it is being made. So now let's look at the steps involved in DNA replication. In the first step of DNA replication, we're going to have helicase, and helicase specifically is an enzyme that is going to unwind, in other words, separates the parental double helix strands. Then we're going to have single strand binding proteins stabilize these unwound parental DNA strands. Then on step 3, on step 4, we're going to be synthesizing the new DNA strands that are going to be base paired with the parental strands. So understand that DNA polymerase is going to be the enzyme that catalyzes the synthesis of the new DNA strands. Now, these new DNA strands that are outlined on step 3 and step 4 are going to be made in different ways. So, when DNA polymerase makes these new strands, is always, so DNA polymerase uses the parental DNA in the 3' to 5' direction as a template. Template means that that is what's going to be guiding the synthesis of the new strands. So what does this mean? Remember that DNA is anti-parallel, which means that we have a strand that is going to be in the right direction when replication happens, but the other one is going to be in the opposite direction, But remember, synthesis has to occur reading the strand 3 to 5. So what that means is that there's going to be one strand that is made continuously, and that strand that is made continuously is called the leading strand. And as you can see, is using the 3' to 5' direction, okay, as a template. And the DNA polymerase can make that the leading strand continuously. But when it comes to the other strand that needs to be made, even though it's being read in the 3' to 5' direction, because that is going away, okay, from the unzipping, then that has to be made in little pieces. So the lagging strand, which is the one that is made discontinuously, okay, is the one that is going to be made in small pieces, okay? Understand that the place where the unzipping happened, that is known the replication fork, okay? Because that's where everything is coming apart in order to synthesize these brand new strands. Now, remember that the lagging strand is going to be made in pieces, and these pieces have a name. They're called Okazaki fragments, okay? One of the things that it is written but not specifically mentioned in the steps, DNA replication requires an RNA or aRNA primer. So what does this mean? That there is a little bit chunk of RNA that is added, and that is what's going to tell DNA polymerase, hey, this is where you're going to start. So understand that this RNA primer is going to be added in order for DNA polymerase to start the synthesis of these new strands. And in the last step of the reaction, that RNA primer is going to be replaced by DNA, okay? And there's a different DNA polymerase that does that. And then we have a molecule that is called DNA ligase that is going to be linking those Okazaki fragments that are found in the lagging strand to make the whole new strand, okay, that is synthesized in little chunks. And remember that that is the one that is called the lagging strand. Now we're going to explore the central dogma of genetic information, okay? So understand that the genetic information or the flow of information goes from DNA to RNA to protein. So proteins are actually made from a DNA strand. Again, when we have the conversion of DNA to RNA, that process is known as transcription. And then when we have RNA converted to protein, that is done through the process of translation. We are going to be exploring now the steps for each one of these processes. Now, understand that when it comes to RNA, remember RNA is another type of nucleic acid. It is actually made from DNA. RNA, interestingly, makes most of the nucleic acid found in the cell. So on each cell, even though DNA may be the longest nucleic acid, most of the nucleic acid in cell is actually RNA. So RNA is the one that is going to be transmitting the genetic information needed for cell operation, and RNA is the one that is converted to protein. Now, understand that the molecules are polymers of nucleotides that are different from DNA molecules in these 4 ways. Number 1, the sugar, as we described previously in RNA, is going to be ribose, and in DNA, remember that is a deoxyribose. When we are making RNA, understand that the base pairing that is needed to synthesize this new strand of RNA is going to have uracil instead of thymine. So as we're explaining the process of transcription, you will see how that plays a role. Also, RNA molecules are single-stranded, and as we saw in DNA replication, after replication, we actually have two double-stranded molecules. After the process of transcription, we have a single RNA molecule. And lastly, in terms of size, RNA molecules are much smaller than DNA molecules. So there are a number of RNA molecules that are present inside the cell, but we are only going to focus on the ones that are on table 17.3, which are the types of RNA molecules found in humans. So understand that when it comes to RNA, these molecules of RNA are called non-coding RNA molecules because they are not translated. So the non-coding RNAs that we have are going to be ribosomal RNA and transfer RNA. So they are obtained by the process of transcription, and that is the final step of the reaction. So understand that even though these three molecules are involved in the synthesis of protein, only ribosomal RNA and transfer RNA remains as an RNA molecule. mRNA is the one that carries the information further and is utilized in order to synthesize proteins. So now let's look in detail at the process of transcription. So when it comes to the process of transcription, remember, DNA transcription is where we go from a double-stranded DNA molecule and we make a single-stranded RNA molecule. The processes of DNA replication, transcription, which is what I'm going to be talking about now, and later when I talk about translation, the explanations are going to be based on systems in bacteria. The reason why we explain the bacterial systems instead of the human cells is because in the human cells are more complex, and these are well-studied systems. Now, let's get started with DNA transcription. So when we have DNA transcription, understand that the first thing that needs to happen is that the RNA polymerase molecule, meaning the enzyme that catalyzes the RNA molecule, needs to identify where in DNA do I need to bind in order for transcription to occur. And that occurs at what is called a promoter sequence. So the promoter is the place where RNA polymerase binds, okay, in order to get transcription started. So as you can see, once the RNA polymerase molecule binds to the promoter, what this is going to do is uncoil, basically unwind and separate the strands of DNA. Then what we're going to have is that in the body or in bacterial cells, which is the system that we're explaining here, we're going to have RNA nucleotides are going to be aligning specifically using, okay, as a template, let me highlight it in a different color, the 3' to 5' strand of DNA. So understand that the synthesis of both the new strands of DNA or the new strand of RNA occurs always in the same direction, in the 5' to 3' direction. So that means that as a template, we must always use the strand that is 3 to 5. So when this happens, as you can see, we're going to have a free RNA molecule or nucleotide that is going to align, is going to be added to a new strand of RNA. And that's when the synthesis of RNA begins. Understand that that RNA polymerase is going to move down the DNA and is going to elongate that polynucleotide for RNA molecule. So understand that in the process of elongation, the RNA polymerase molecule is going to move down the template, which is the 3' to 5' strand of DNA, to make that RNA molecule. So as you can see, the reason why this happens is that this RNA molecule that is newly synthesized is going to be guided by the base pairing with the 3' to 5' strand on DNA. Then understand that this process stops at what is called a termination site. And in this termination site, what is going to be occurring is that other proteins come and tell the RNA polymerase molecule, you are done synthesizing RNA, so you can just fall apart from this DNA molecule, and then the RNA is going to be further processed. So once we have the molecule of RNA made, understand that RNA is going to undergo the process of translation. So if we relate this, at least to human cells, I should specify that the process of DNA replication occurs in the nucleus of human cells. The process of transcription occurs in the nucleus of the cell, while the process of translation is going to be occurring in the cytoplasm of human cells. Okay? Now, in the process of translation, remember that this is where we take that RNA molecule and we're going to be turning it into protein. So, whenever this happens, understand that the components to make this process occur is because we have a tRNA molecule, which is what's going to be carrying the amino acids. We have the machinery, which is called a ribosome. And this ribosome has a large subunit and a small subunit. In addition to that, we need the mRNA, which this is the messenger RNA that was made in transcription. In addition to that, we do have a ribosomal RNA. So if we think about it, the ribosomal RNA, I'm just going to draw it in. It is part of the ribosomal subunit. So those lines that I drew inside the the large and small subunits are going to be the rRNA. It's just that it's not illustrated in this figure specifically. So these are the steps for RNA translation. On the first step, we call them initiation step. So during the initiation, understand that the components for translation are going to come together and bind at the mRNA. Specifically, the first amino acid that is always incorporated onto a protein is going to be methionine. Because as we're going to see in the genetic code, that is the start site for all protein synthesis. So as you can see, the mRNA is going to be sandwiched in between the two subunits of the ribosome. The tRNA is going to identify the start site what is called the start codon. And to specify codons is going to be 3 letters, and remember that those letters are because of the nitrogen base, that are going to identify what is the amino acid that needs to be incorporated. So that's what is called a codon. So methionine is the start site, and that's the first amino acid that is incorporated. During the elongation process, understand that amino acids are going to be bound to the mRNA by these tRNAs, and they're added one by one to the growing polypeptide chain. As you can see, when it comes to the tRNAs, they are going to be identifying, or they're going to be identified, I'm going to highlight it in green, by reading codons. Oh, that's hard to see. Let me try green. So here, it is a little bit hard to see. Maybe I'll use my pencil. So here, what I circle in magenta, the tRNAs are going to be identified codons in the mRNA, and that's how it's going to know that the correct amino acid is incorporated, okay? So remember, the tRNAs is going to complementary attached to the codon. So this is going to be giving the tRNA codon, and this is in the mRNA. They're going to be base pairing with each other. I'm just going to symbolize the base pairing by this highlight in green. And that's the interaction that I circle in magenta. So during the termination process, understand that we have specifically a 3-letter code in the mRNA that is called a stop codon. And the stop codon is where specifically this molecule that is called a release factor is going to bind and is going to allow the completed polypeptide to come away from the ribosomal machinery. So understand that the steps in protein synthesis are all outlined in table 17.5. So as you can see, the steps that you guys need to remember is that first we have DNA transcription occurring. Then the tRNA, which is what brings the amino acids to the molecule, is going to be activated. Then we're going to have elongation and initiation, and chain elongation, specifically for making protein. And then we're going to be terminating specifically the synthesis for that protein. So as you can see, the steps that are outlined on table 17.5 include both the process of transcription and translation, because this is giving you the overall view for the flow of genetic information, meaning how DNA gets converted to RNA, and RNA proceeds to be converted into protein.